Welcome, and the floor is yours.
All right, great. So thanks and welcome everyone. Yeah, my name is Andrea Schnepf and I'm really happy to be here. Thanks also for the organizers to invite me. And I believe my talk should have been on one of the earlier days where the theme was something like life, I think. So I was asked to present about the role of plant roots that are in the soil in the hydrological cycle. And I will do that, so the work I will present is kind of a synthesis of the work of our group and also embedded in the corresponding community, and I will refer to the corresponding relevant publications wherever it's appropriate. Also, I want to present the work in such a way, I hope it's in such a way that in the end you will have tools for yourselves to create your own root architectures and your own root water uptake sink terms and do some computations. So, and as I also had the information that plants in general and root probably in particular are more a side topic of this whole summer school, I wanted to start with some general motivation. And so here, just to also say that roots have made it into proverbs and quotes by different famous people. And also general proverbs in different communities. So people had, for different reasons, something to say about the roots. So if we also look at the estimates of the global biomass carbon, then there are also estimates that of all of the biomass that's distributed among all of the kingdoms of life, 30% of it is in the roots. So we can expect they kind of probably could have some important role. And I want to look at one in particular, namely the role of the roots in the terrestrial water cycle, namely the transpiration, because the roots are the place at which the water taken up from the soil has to pass through to enter the atmosphere through the plants and through the leaves. If we look at it on the scale of a global water cycle, we can also see that the evapotranspiration flux from soil into the atmosphere or from land into the atmosphere is rather large. So we have 73,000 cubic kilometers per year of water that moves from the terrestrial land into the atmosphere. And there are different estimates how much of it actually passes through the plant. So either the water moves directly from the bare soil through evaporation into the atmosphere, or it moves through the plant tissue and through the leaves into the atmosphere. And there are also estimates that it's at least half of it that passes through the plants. Yeah, and so you can look the data up from this publication from Trenberg. Also, this flux, if we look at it throughout the year, is not a constant flux, but it also varies over time, especially for agricultural crops that have a vegetation period, for spring crops. So what I plotted here for you to see is the time on the x-axis, which would be the vegetation period of a maize crop from planting to harvest. And then you see the crop evapotranspiration in black and how it's distributed between evaporation from soil into atmosphere and transpiration through the plants into the atmosphere. And so you see in the beginning of the season, of course, almost all water moves directly through the soil into the atmosphere, and then later the plants pick up. We can also compute this crop evapotranspiration with the Penman-Monteith equation if we know the climate data and the crop coefficients. And then we can use Beer's law to partition. So the computation here that's plotted here was partitioned the potential evapotranspiration into the transpiration and evaporation part using the Beer's law, and that's based on the leaf area index. So how many leaves already have developed at a specific point in time. And here I try to put together some numbers. So here I just visualize that the water moves from the soil into the root, are transported upwards, and then leave the leaves into the atmosphere. What's that in numbers? So I tried to bring them together for an example for a maize plant and an oak tree. And so we have on a daily basis, one maize plant approximately transpires 1.8 liters per day, while an oak tree even 400 liters per day. And if you divide this just by the root volume, you can see that it's quite a lot of water that's basically moved. So the volume of the roots is flashed several times a day just by the amount of water that passes through the plant. So it's a lot of more water that's actually needed by the plant itself. So how are we gonna describe this process? And so here the concept of the so-called soil-plant atmosphere continuum is important. So where the water moves along a continuous column of water movement and moves according to potential gradients. We are here put the numbers, so you already see in the illustration that I will focus on the roots, so they are quite detailed, while I will not put the focus on the above ground part today, so it's just one lumped leaf. But you see the water potential in these different compartments, very negative in the atmosphere, less negative in the leaf, and so on until we reach the soil. Notably, it's all negative. So the water inside the plant is under tension. And how much is it? So here we have minus 1.5 megapascal as the tension in that a plant could experience. If we make another simple computation, to say what would be the minimum leaf water potential if we wanted to lift water 100 meters up against gravity. What would we need? What would that mean? And so we can simply, or do you have a guess?
1 megapascal.
Yes, exactly, because what you just need to do is you just convert these 100 meters in head units to the pressure units. And here you can compute it and you will see that in the end you will end up with minus 1 megapascal, which is kind of a relatively strong negative potential which the plant has to also sustain. And also here I found some literature about this topic, and what you can see on the left are leaves on a sequoia tree, and the numbers on each of the leaves are the height at which they were sampled. And you can see that in the bottom they have broader leaves at a wide angle, and as you move up the tree they become narrow and small, and so basically trying to reduce the openness of the stomata and trying to reduce transpiration. And so there are then the ideas that this is basically a physical limit to tree height, because at some point there is a carbon starvation. So you cannot no longer, if you close your stomata, you will decrease your risk of cavitation and other problems related to this negative potential, but you also then close your stomata. That means there will be no photosynthesis and no carbon, and so no more plant growth. And so that could be a theoretical limit to the height of a tree. But now I want to come to the actual topic. So the root architecture. So let me start with the root architecture. Let me first start to take my time, because otherwise I will bore you for too long. So you can enjoy this nice image. Okay. And basically we distinguish taproot system and fibrous root systems, where there is so in the taproot systems in red on the left you have a main root, one main root, and it creates lateral branches and more lateral branches. So that can go, there's some kind of self-similarity in it, so that's why root architecture models have also used L systems to simulate these. And fibrous root systems, you have additionally so-called shoot-borne roots. So there are nodes on the shoot which can also develop roots that will then grow into the soil. That will be the secondary root system, for example, of grasses. And if we look at it on the whole root system scale, they also come in different forms and shapes. Here you can see some examples of different plants. And you can also see that for some of the plants, the below ground is actually much larger than what you see above ground. And if you are more interested in it, there is a book by Lore Kutscherer called Wurzelatlas. So it's like a root atlas, and it's a 7-series book where Lore and her colleagues have excavated, like archaeologists, by hand these roots and made drawings, and for all kind of different species. So you can really get an idea for different plant species, crop and wild plant, how the roots look like. And they have been digitalized in Wageningen, and the collection can be found on this link you see in the bottom here. So in my group, we developed a model called C-Plant Box, which tries to create this root architecture, or that was our start, as realistic as possible. In the meantime, it has evolved into what we call a functional structural plant model. We also simulate the whole plant architecture, including the leaves, but I will focus on the roots and water uptake today. And the core code is is in C++, but we have Python shared library, so you can use it with the knowledge of Python. You can create all the structures by yourself. And the GitHub repository you see is open, so anybody can use it. Also on this GitHub repository there are some tutorials in Jupyter notebook form, so you can just try them yourselves. There is also some published model root architecture parameters available, so you can select the kind of plant you want to plot and create your own root architecture. And you can also find examples of different applications, including not only the creation of the structure, also the growth of the plant inside a container that limits the plant growth in certain dimensions, but also then functional modeling, carbon and water flow inside the plant, and water and nutrient uptake from the soil. We have linked it to different other available codes that operate on a larger scale. For example, Simplace is a crop modeling platform developed at the University of Bonn, and PF Münch is developed by a French group that simulates the Münch theory, coupled water and carbon flow inside the plant. So we can couple our code to all kinds of different other models depending on our applications and research questions. And for more interest in the above ground plant parts, you can refer to this publication of Giraud in 2023. And now I want to show you how coarse and simple the root architecture model really is. So we start with one seed, and the root growth is elongated. The root is elongated, so the meristem where the new cells are formed are only at the root tips. And so in each iteration, the root tips can elongate. The root can elongate itself, mostly along this equation here, where the root speed of root elongation becomes smaller as the root ages, so with time and as it reaches its maximum length. So then the roots can elongate, and while it elongates, within the branching zone it forms new lateral roots, which are now indicated by these new meristems as the red dots. Yeah, and they also elongate, so in each iteration each meristem elongates and forms more roots. As they become large enough, they again can form laterals and so on. And we also have to define the orientation. So in each iteration, our root tip can also decide, do I want to change my growth direction? And by this, we can implement different kind of tropisms. For example, the root should grow towards downwards, or towards, for example, water patches, or any kind of tropism we would like to implement. The bottleneck here is the parameterization of this tropism. So how we do that is, if we come from an old growth direction, we have two angles that will determine our new direction, and one is how it's changing laterally, and then how it's positioned around the root in a new direction is this second angle. So this is what I try to illustrate here in blue and green. Yeah, and this is different examples from our paper where you can see that we can create all kinds of different root architectural structures with this code. The colors here indicate the age of the root segments, so we can see that, for example, if you look here, you can see well that the oldest part of the root is near the stem, and the youngest part are always at the tips where the meristem is. So at the tip itself, the root has age 0. At the base, it has its age as long as it has been existing. Yeah. And then here are some visualizations, what else we can do. So we can grow the roots inside containers. Here we have a cylindrical container on the right, and on the left a so-called rhizobox system where people who do experiments grow them in pots that have a transparent window and then are grown at an angle so that the roots grow along this transparent surface. So with this code we also try to mimic different experimental setups and then learn from other interpretation. Here you can also mimic field experiments with it, so we can put several seeds at the plant and inter-row distance as it's done in an agricultural field, and then we can simulate the root development over time. So what you also see, these blue tubes here, that's a visualization of an actual field site we have near Jülich, where we installed horizontal plexiglass tubes in the soil, where we can put endoscopic cameras in it and also then observe the root growth over the whole of the vegetation period. Also you can put other things in it like GPR antennas and do geophysical measurements. So don't go into these details for today, but this is also some experimental work we're doing.
Quick question?
Yeah?
So maybe you will discuss this, but so what are the rules of interaction for root growth from different plants? How do they feel each other in your model?
So in our model, they don't explicitly feel each other, because usually so we have focused so far on one individual plant, not so much on interaction, although that's in our mind, of course, that we should do that. How they interact currently is indirectly. So for example, if we have root water uptake, and we have several root systems, then they will all take up water from soil, so root will sense then if the water is already dried out by another plant or not. So indirectly, roots will sense that. Directly, I don't know if we exactly know even the mechanism. So we could, of course, implement. We have also thought about implementing this. Should we have some kind of like a magnet or so that they attract or oppose each other? But there, so I really don't. I'm not sure if we know the exact mechanism of direct sensing of other roots, although I know that there is work that shows that there are differences. For example, if it's a friendly plant or kind of one plant that is not so liked by the plant. So but so this is not part of this current code, only indirectly. What we have done also is we've spent some time to think about data structures, how to store the information of the root information is basically stored that we have points in three-dimensional space. So each point has 3D coordinates, and then we have information which points are connected with each other, and then this is kind of our information about the root architecture. And then we can also add additional information. So for each root node, we can also add its radius, its age, its different hydraulic properties, information that we might want to add. And so we developed this XML-based data format called RSML, Root System Markup Language, which is now kind of relatively widely used, where we have a specific nested way to store this information. And I just wanted to say that this is important because in the meantime, there are several tools that use this data structure and they can interoperate. And this is, if we have, for example, data from such rhizobox experiments, we have same data structure, and then we have models, experiments, and other kind of analysis tools. They can all read or write RSML files, so it makes it easy to use, for example, experimental data in a modeling exercise. And this information you can find, if you're interested, in this publication by Guillaume Lobet in Plant Physiology. So now let's move to the next topic. So now let's say we have now our structure. It's described in the way I said. So it's a discrete representation of our root architecture. We have points that are connected by root segments. Now we could say, okay, we can now describe relatively simple the water flow at the root segment level. So we have two kinds of flows to consider. One is for each root segment between two nodes. We can describe the axial flux or volumetric flow rate by its difference in potential at each end of the segment times the conductance. So we have the driving force is always a potential gradient. And then how well or not well, how resistive is my root against this water flow is described by our conductance. It's basically the 1 over the resistance is our conductance, and it tells us how easy it is to follow this flow. But the driving force is always our potential difference. And it's actually the potential gradient, but the distance over which the potential occurs is now hidden in this representation in our radial and axial conductance values. For the axial flux, it would be the length of the root segment. And then for the radial flux, it would be the root radius. So the distance from the soil into the xylem. What is the driving force for the radial flux? So that's now the water that could potentially move from soil into the root. Here we have to look at the water potential difference between the soil at the root surface and within the xylem. So it's a rather simple description on a segment level. It's a little bit confusing, maybe if you look in the literature, that you will always find such strange units and different units in each paper. It's really painful sometimes. And this also has to do with the fact that we have 3 different water potential kind of units, right? If we have pressure unit or length unit or joule per kilogram units. And so depending on what unit you use for your water potential will affect the unit you will have for our conductance. So this is an easy way to make mistakes if you're working with different sources of literature. So what we can then do is we can now approximate our water flow as like a finite difference approximation, really. So I put on the left like a simplified root architecture. We have one main root and it has one branch. And each node now has a number and each segment has a number. And we can collect the information which node is connected with which each other node in this so-called impedance matrix. So we have, for example, in the first line, there's a water or there's a segment going out from node 1 and into the node 2, and so on. So you can do this for all of the nodes, and you can collect just the information of the connectivity within a relatively simple matrix. And then you can use this information so that if you multiply now this connectivity matrix with a vector that contains our variable of interest in this example, the water potential, then what you get is basically just a vector of the differences of our water potential in each segment, each root segment, across each root segment. We can then multiply it with this diagonal matrix where we collect all of the information of our conductances for each segment, and then we end up with this kind of Darcy flow-like water flow through the xylem, which is driven by the water potential. And then we can do yet another matrix multiplication, namely the transpose of this connectivity matrix. And if we do that, we end up with another vector where we compute for each node the sum of all the in and outgoing fluxes of water. So this is basically nothing else than Kirchhoff's law, where we collect how much water flows in and out of each node. And that's kind of a relatively numerical, discrete representation of our water flow through this system. If you're familiar with the solution of partial differential equations on a 1D discretized domain, you will recognize that we can represent our partial differential equation by this system of equations where we have our Laplacian times our vector of variable of interest, and this equals to some right-hand side, which we haven't talked about yet. What this L for a 1D domain looks like is just this tri-diagonal matrix, if you are familiar with it. And on the right-hand side, we would have two boundary conditions on the top and on the end, and we could have here on the right-hand side some sink or source terms, for example, which we have not. So if we do the same with our root architecture, it's really the same. Only our matrix looks a little bit different. We have also some off-diagonal entries according to how the root architecture is branched. And then we have for each root tip its boundary condition. Generally, we prescribe zero flux at all of the root tips, and we have one special boundary condition, what we call the root collar, where we prescribe the transpiration flux. So here, from here, we pull the water, we prescribe how much water should be pulled from the root. So now we can then discuss what we put on the right-hand side of this equation. In addition to our boundary condition at the root collar, where we prescribe the water flux, we can say on the we describe the water movement from soil into root by this radial water flow terms as a sink or source term into our Laplacian, into our graph representation. So what we have is this, you probably remember our equation from the beginning. We also collect this in this vector form, and this is basically our quite simple system of linear equations which we can solve for Hx. So we can solve it for the water potential at every node in the xylem. If we know the soil water potential, and this is not always the case, we can then easily solve this as a linear system of equations. And then with some algebra, which basically I don't want to go into the details, but you can look it up in this paper by Jan van der Borcht in 2021. And the basic idea of it is we actually solve this linear system of equations for the water potential in the xylem. We plug it in here, right? Then we have a description of our water flow, root water uptake, that doesn't explicitly consider our xylem potential, so we don't need to explicitly know it. And this makes it easier for the coupling to any soil models. And then some further algebraic formations. You can then come to this equation, namely we have two terms, KRS, the effective root system conductance, times the SUF, the standard uptake fractions, times potential difference, which is the collar potential at the root collar. And then we have the effective soil water potential, which is the SUF averaged soil water potential. That means the average water potential the root feels, basically. So So we have a scalar here. So, and if we for a moment do not consider this term, then this means we have a vector standard uptake fraction which is kind of similar to a normalized root length density, only it also includes root hydraulic properties. So it distributes what the plant should transpire to the different soil layers, or to the different root segments here according to this vector. Now if for a given position of a root there is a difference to the average one, so if the soil water potential is not uniform in the soil, then there could be a difference from a given location to the average one. And so then we can observe a phenomenon naturally comes out of this description of this equation, namely the compensatory root water uptake. Yeah, so if we have a region in soil which is drier and there is less water uptake, it can be compensated for if the other parts of the root are in a region that are wetter. So this comes out of this formulation. We also have implemented in our different tools like C-Plant Box and also Marshall, which was developed by Félicien Meunier in UC Louvain. So some tools compute these functions and this scalar for you, and they are basically intrinsic characteristics of our root architecture. But now instead of having this huge matrix and all of the 3D position, we have only a scalar and a vector by which we can describe our root architecture, hydraulic root architecture. Now this information now is per root segment. So it still kind of could be rather large computation. We could have a full grown root system could have 60,000 segments or so. So it's not very small. So if we can find patches of soil in which the soil water potential does not vary, we can then upscale our formulation by just summing up basically these different parts according to so this now here is illustrated for if we have different soil layers. So here we would kind of map all of the root segments that are within one soil layer in that one soil layer, and that means we significantly decrease our degrees of freedom, because we don't no longer have maybe 60,000, but if we have a soil model that has 1D layers, maybe we have only 100 instead of 60,000. So that's one way to decrease our our computational demand. What we then though assume, that's a simplifying assumption, is that also the xylem potential and the specific potential at the soil root interface are the same for all of the root segments within one soil layer. So that might not be necessarily true. But now we have this root water uptake per soil layer, and that was another step that we did towards finding a formulation that can be implemented into other models that have, let's say, 1D soil modules. Now we have to talk about the root hydraulic properties, axial and radial conductance, which we need to somehow come up with a number. And so here on the left you can see anatomical slices and for the example of a maize root, and you can see that it varies from the basis of the root towards the tip of the root, and it varies for the different root types. So lateral roots, of course, look different than the main root and so on. So they have different diameters, different xylem vessel numbers. Some of them have a Casparian strip, others not, and so on. And based on this anatomical information, we can compute the axial and radial conductances. And we can see, first of all, that as the root ages, the radial conductivity decreases and the axial one increases. So the roots turn from places where they take up more the water towards one where it's transported. So how can we use the anatomical information to compute these? So here you can see on the left-hand side an anatomical cross-section of a maize root. And these circles in the middle are our xylem vessels. Those are the ones in which the water flow axially occurs. And we can approximate this by using Poiseuille's law, which takes into account the radius of the individual xylem vessels to the power of 4, and also the viscosity of the water to compute, it's basically laminar flow in pipe approximation of the water flow in the xylem. Now, for a given anatomic structure, this would then be a constant, right? Because the structure, it looks like this, we can only have a constant root axial conductance. Of course, we also know that within these pipes, because they are under tension, or not pipes, but within our xylem vessels, because the water is under tension, cavitation might occur. And this occurs, the higher the pressure, the higher the water potential, the more negative it is, the more likely is cavitation to occur. And so we can have kind of a non-linear relationship of the xylem water potential and the specific axial conductance. Here I found this example for a maize plant for different cultivars, a drought-resistant one and a drought-sensitive one. And you can see the drought-resistant one relatively stays at a certain value until our magic number of -1 megapascal, and then if it becomes more negative, also it gives up, so to say, and can no longer transport water. The drought-sensitive one starts to cavitate already earlier. So this is kind of one source of nonlinearity in our system. Then we have to talk about the radial conductivity. So when the root passes from the soil into the xylem, it has to move past different tissue layers. So the innermost one is the stele that contains our transport vessels, the xylem and the phloem, which is surrounded by the pericycle. This is the layer of tissue at which lateral roots can be created. And then comes the cortex, which has the endodermis and the exodermis as at the start and at the end. And then the outermost layer is the epidermis, where also root hairs can emerge. And you can, if we know a cell-specific root anatomical representation, we can, Valentin-Couvreur created this mecha model, which computes the water flow through these layers of tissue. So he discretized this and he computes, here this is basically a scheme how he discretized our root. We have the symplast here, and we have the symplastic pathway of water, which moves from one cell to the next via these channels that are via the plasmodesmata that connect each cell. And then we have, because it's a plant, we have also the cell walls, and we can also have water moving within the cell wall, and that's the apoplastic pathway. Also, we then in the apoplast, we at some point reach the Casparian strip. It's kind of a waxy lignin-based structure which omits water flow. It's a barrier for water flow. At this point, at the latest, the water has to do a transmembrane movement that goes from the cell wall into the symplast and then from there into the xylem. So I don't want to go in all of the details. I just want to let you know that based on these computations where we can compute the flow path through all of the cells and parts of the cells, in the end we can compute one number, namely our root radial conductance. And this is the one we then use for our root architecture level computations. If you want to know more about the wondrous paths of water through all of these cells, I refer you to this paper of Valentin-Couvreur, and he illustrated it with this image, I think by Escher, an artist, where the water at all points seems to flow upwards. And so here you will find a lot of the details of how the water explicitly moves through all of the different cell parts. Now we have values we have a root architecture, we have values for the axial and radial conductances. Now we have to also add the soil into the picture. And we do that by computing sink and source terms. So I illustrated this, so the soil is here illustrated by this box. And then we can map all of the root segments into each cell of our discretized soil domain and compute the water uptake from it. Yeah. And so basically what we can then simulate is also water flow towards each individual root segment if we want. We can get from this simulation is the 3D position of the roots, the 3D position of the water content, potentially also solutes, which I didn't talk about today, but solute concentration, 3D information about all the fluxes within the soil, within the root system, and across the membranes. And here now, then we have to think about the fact that the soil is not just given and also not static, but we have a dynamic soil. So here you see an example where the root of a simulation in which we have a growing root, and the root takes up water, and as it passes through the different soil layers, it dries out the soils. And so I assume that you have heard about the stomatal regulation and about the soil already earlier this week, so that at least was my information. So I don't need to discuss the Richards equation, so we solve the water flow in the soil by the Richards equation. And then in each time step solve this linear system of equations for water flow inside the roots, compute the sink term for water uptake, and then we continue the simulation. So the exchange of water between soil and root domain is realized via the source and sink terms. Why do I say source and sink terms? I have only mentioned so far that the water moves from soil into the root, but really it can also move the other way, because the driving force is only our potential gradient, so it could have different signs. And that's what is called hydraulic redistribution, and I can show you a little bit of it later. Now I want to mention some sources of nonlinearity. The second one, cavitation, we have already discussed. I'll put another citation for it. And the other one is what we call the perirhizal resistance of water flow towards the root. If we want to know more about it, you can also look it up in this paper of Jan van der Bocht in 2023. To motivate it, I will also want to refer to you to this paper of Andrea Carminati and Mathieu Javot in 2020, where they picked up this. So there is apparently an ongoing debate in the plant physiology community. So what is more important in regulating the stomatal conductance? Is it the xylem and cavitation and therefore the loss of water flow through the plant, or is it soil resistance? And so Andrea and Mathieu came up with this figure where, so they call by E, they mean transpiration. So there's some part of community that uses also the E for evaporation but when they mean transpiration. And then you see the soil water potential on one axis and the leaf water potential on the other axis. And you can see these curves here. And they mean if for a given soil water potential, if I want to have more transpiration, how much more negative do I have to get in my leaf water potential? And you can see that for some time it stays linear. So I get a certain reward if I am, as a plant, so to say, willing to accept more negativity in my xylem potential, get a certain reward, more transpiration. But then the curve starts to bend, and I have to, for the same amount of transpiration, I have to get much more negative. And so they found this red curve here, which is the curve where they could also show by a meta-analysis of different published data that most plants tend to stay in this domain here and rather not go into this brown domain. And that has to do, why does it bend? This has to do with the fact that the soil becomes in the rhizosphere much more negative, and the soil hydraulic conductivity drops to a very low value, and water is no longer transported into the root. So we tried to put this into our model, and how we did that was, here you can see on the left a root architecture, and so we divide for a given, so for each root segment we compute by using Voronoi polynomials its available volume. So for each root gets its own little volume, so that the neighboring root, there's always a half a distance of space between all of the next neighboring roots. And so from this we can then make a simplification, namely we assume it's really a radial volume of soil which is available to our root, and then we can compute at the radial distance from the root axis for a given transpiration what's the soil water potential. So I forgot here the y-axis, so that's our water potential, and we see that as the soil dries, the soil water potential at the root soil interface very close to the root surface drops dramatically. So it really becomes very limiting to water flow. And we then used an analytical solution using the steady rate approximation, which is described in a Schröder et al. Paper, for a 1D radially symmetric description of the water flow using Richard's equation. And you can compute the water potential from from the bulk soil towards the root surface according to this analytical solution. And we always know our bulk soil water potential. We know our xylem water potential, and we then can compute with our analytical solution the real soil root interface water potential, which could be much more negative than the one in the soil, in the bulk soil. And then also using, so the analytical solution is based on this metric flux potential linearization of the Richards equation. And I just want to point out that this one here is now our important parameter, the perirhizal zone hydraulic conductivity. And then we have another factor B, which is a geometric factor which depends on the radial distance of the root surface, so the root radius, and the outer boundary of our radial soil domain. So basically now we have a nonlinear and also implicit system of equations which we need to solve, because here our perirhizal zone hydraulic conductivity depends on both the xylem water potential and the root soil interface hydraulic potential. So we can do some iteration. We have to use some iteration scheme to solve now for our solution. And now this is a description which is again based on the root water uptake per segment level, but the upscaling to the soil layer level can be done then in the same way as we have done before. Here I wanted to also show you some examples of 3D simulation setups where we mimic the root water uptake of plants within a crop stand, and we do this by having a soil domain which has the horizontal dimensions are the interrow distance and the plant distance, and it has some certain depth, let's say 2 meters. And then we put on the sides of our soil domain periodic boundary conditions, so that we basically have one representative plant in a crop stand, and then this is kind of repeated periodically. And this is then how this simulation would look like: on the left for a spring barley crop stand, on the right for a maize field, where we have these distinct row effects. And then we take out this one box and do the simulations on this one box. You can also see the interrow spacing and the plant spacings are quite different between these two crops. And this is then an example how such a simulation output looks like. So we have on the left the 3D soil domain and a 3D root architecture, and you can see the colors indicate our soil water potential, and we can see where the root has dried out the soil. And then on the right hand side you see the effect when we make a simplifying assumption that our soil domain is actually not 3D, but we assume it has only 1D layers. And then we can see that, of course, that all of the lateral heterogeneities are gone, and we work with an average value per soil layer. So, yeah. So then I also wanted to let you know that the way we computed it was also part of a benchmarking exercise. So we collected different groups who develop similar kind of code. And we compared our simulations to a reference solution, which was an explicit 3D reference solution, where we actually discretized the soil in such a way that all the roots are basically holes in the soil domain. So it's no longer a simple volumetric sink term root water uptake, but really discretized soil all around the roots and all the faces of the roots explicitly considered. And we compared it to this root water uptake sink term approach, because obviously it's computationally much more demanding if we actually do the explicitly 3D simulation. And of course, when we have to compute our root water uptake sink term, we have different possibilities how to distribute the sink term back into our soil volume. And we have also different methods how to determine our soil root interface water potential. So the one I just showed you is one of several options, and the choices we make here have an effect on our numerical outcome, and we wanted to know how big is the numerical outcome. And so I guess in the end we grew somehow to converged towards ways how we did it, how we wanted to do it. Yeah. Then here I also wanted to show you as an example just a theoretical computation. So the setup was we have a soil profile which contains roots and which is very dry at the top and rather moist at the bottom. And we wanted to kind of put this idea of hydraulic redistribution to an extreme. Yeah, we said, okay, we make a simulation where the roots are there, but they are never transpiring. So we always it acted as if the roots were always due at night and have a root color potential of 0. And so they are just acting as kind of hydraulic highways in the soil, but not actually really transpiring anything. So the soil is not dried out, but the water is redistributed in this simulation. We had also no flux conditions at the top and at the bottom, and just wanted to wait how long does it take until we reach equilibrium water distribution in the soil. And on the left, when we had only the soil and no roots, it took 10 years until the equilibrium was reached, theoretically, hypothetically. And on the right, when we added the roots, still no transpiration, but the roots acted like wicks in the system. Then it took only weeks until we reached equilibrium in this simulation. It was just some in silico kind of experiment to see how strong it can really be the effect of roots. In reality, we would have a situation where at night there's transpiration pull, water moves into the roots mostly from the soil, but at night the transpiration ceases, or it becomes at least very small. And then we have this hydraulic redistribution occurring, where water is taken up from moist parts of the soil, released back into the dry parts of the soil, with an overall net 0 uptake. Yeah, it's just really a pure redistribution. And this is then how it looks like. Yeah. So now we have the sink term the syncterm depth distribution, where on the left water is taken up and on the right it's released back into the soil. If we compute the integral on these curves on the left and on the right side of this zero, we should have the same number. Yeah, so then we have, then we wanted to go one step further and say, okay, well, this is all very nice. Our water moves according to potential gradients and so on, but it's still rather hard to get other people like crop modelers or land surface modelers convinced they should implement this. And so we said, okay, we should go one step further and made some further assumptions, simplifying assumptions, namely that the structure of the root architecture is not as we saw it before, but it is the so-called parallel root system. So we have a root collar at the top, and then each soil layer has one root that is directly connected with the collar, so that all the roots exist in parallel in our soil domain. And we also said, okay, the root collar potential is always at the critical, minus 15,000 centimeters. When we do this, we can come up with a sync term which is really simple and which most of all doesn't need any iteration anymore. And it is this term, so we have the easiest one is the top equation. You remember the standard uptake fraction term, and then our transpiration pull is what we prescribe. And then we have only two additional values, namely our alpha and our omega variables, parameters, which I will describe on the next slide. Alpha and omega, if you are familiar with root uptake models, are relatively well known. So there is a FEDES, so-called FEDES approach that uses the alpha factor to indicate soil layers in which the demand cannot be fulfilled. So there is a stress factor. And then there's also some formulations which consider compensatory root water uptake via this omega parameter. So we wanted to find a way that looks similar to all of the known or to the well-known root water uptake functions, but can be computed from the knowledge of the root hydraulic architecture, from the full 3D information. And so this is now, so here, so the first one is when omega, which I will describe in a minute, is smaller than a critical value, we use the top equation, which basically does nothing more than just redistributing what the overall volume of water that needs to leave kind of our plant to the different soil layers. And then in the bottom, we reach a situation where we do consider also our soil, with the restrictions through the soil water potential. And so here I put together like a comparison of our new parameters, alpha and omega, to the analog in classical root water uptake models. And so the first one is the standard uptake fraction. And it's rather similar to what is normally done, namely using the normalized root length density, because this is something that has been easy to measure, root length density, and standard uptake fraction is rather similar, but is calculated from the 3D hydraulic architecture, also incorporating our knowledge of our root axial and radial hydraulic conductances. Then we have alpha, and if you know the FEDES model, then you will see it's a soil layer specific stress factor. It's the ratio of the actual root water uptake and the demand from the layer, and that's a function of the total of the potential transpiration rate. Then our alpha here is the ratio of the maximal versus the potential supply from any given layer I, and how much is the possible potential supply depends on our current bulk soil water potential. So it takes into account what is the current soil water potential, and that is the maximum that could be potentially delivered. And so alpha computes this, this ratio of maximal and potential. Then we have omega, which basically only sums up the alpha over all of the layers. And then we have the omega critical, and that's in the classical root water uptake models a critical stress factor, which is defined. And it does not depend on the demand. And in our formulation, it depends on, it's the ratio of the potential transpiration, so what should be transpired by the plant according to the atmospheric conditions, and then the potential supply. So now our omega function is a function also of the demand, current demand. But in principle, it's rather similar. It has alphas and omegas, and the function is also quite easy to implement. And so it makes some simplifying assumptions, and we are now also in the process of quantifying for each simplification how much uncertainty or how much inaccuracy do we then add to our numerical solutions. And yeah, so I'm now already coming to an end. So first, to summarize, what I try to show you is how to compute the root water uptake from the knowledge of the root hydraulic architecture. We can do it in a more complex way, we can do it in a less complex way. I also introduced to you the intrinsic root system characteristics, the effective root system conductance, KRS, and our standard uptake fraction, SUF. And you can compute this for any given root architecture you have. By doing our simulations, we respect the underlying mechanism of water flow along the hydraulic gradients. And that is actually different from classical root water uptake models, because they are very often a function of the soil water content. And of course, somehow, it's also then a function of water content, but it's not the driving force. Really, it's the water potential. We also looked at how to compute the different individual resistances along our water flow paths. And also that the soil, what we call the perirhizal zone, so the soil volume around the root which is influenced space-wise by our root, this resistance to water flow can become dominant in the overall path as the soil dries out. Now, what is still things to do? And so, first of all, we really would, we are now in a phase where we meet different people from the crop and land surface modeling communities to discuss the implementation of this new root water uptake sink term in different models. It's already partly implemented in Hydros, CLM, and Simplace. And then another topic which I haven't discussed is to look at the water storage changes in plants. So all of the computations I showed you are based on the fact that there are actually no water storage changes in plants, right? But at least for larger canopy like trees and so on, we know from measurements that this exists. And then we would have to do some more complex simulations that can account for water storage changes. Work towards this direction in terms of measurements is, for example, done by Alexandra Konings, which you can look up in this paper in New Phytologist. And also in modeling-wise for trees, it has already been described in a group that originally comes from Eckhard Briesack in Germany. Then we also have to consider or we may want to consider plant adaptation to environmental conditions. Here I want to refer you to two papers. One is a recent paper by Pang Yu from the University of Bonn, where he looked at the changes of maize root architecture during the domestication of maize from 8,000 years ago until today, and he could show that because root system has never been bred on purpose until recently, but it has changed for the different climatic regions in which the maize traveled from South America to North America to Europe to China, and he could show a relationship between the climate and the root architecture development. And then Gernot Bodner looks at it more from an agronomic perspective. How can also management measures help for plant growth in different environmental conditions? And so I only hinted on it that with our model we have this tropism term where the root can change its orientation. We really have in our code ways to change the elongation rate, the angle, and the branching angle and the branching rate according to environmental conditions. And we have done also some simulations like hydrotropism and so on. But here the bottleneck is the parameterization. How is actually really all this affecting these different properties of the plant? But in breeding communities, it's quite highly discussed at the moment. Yeah, so is plant plasticity, a trait we should breed for in times where the weather conditions might be unpredictable or less predictable than they used to be? And another topic which I also like to follow is to integrate our above ground and below ground views of the system. Yeah, because often researchers tend to have either they look on the above ground or the below ground, but we really know that the plants are the ones that link the two. I've shown it for the water today. Of course, vice versa for the carbon, it's the same. So photosynthesis, a lot of it is distributed below ground into the soil, and then to really simulate this, we started to do that with Mona Giraud in 2023. You can look up this paper, and we have now extended this by follow the path of the carbon from photosynthesis through the plant, the growth of the plant, the maintenance of the plant, and the release of rhizodeposits in the soil, where they then influence the soil microbial community. So it's really a big modeling effort that we are now in the process of finalizing. And of course, all of the above ground mechanisms you already have heard probably a lot this week. So I guess there's lots of things to interact. And just to finish, so I was also asked, I don't know when this will take place actually, but to prepare a hands-on session. And this is one where I prepared for you some Jupyter notebooks, which you can run through Google Colab. And with those Jupyter notebook, the first one will show you how to simulate the root architecture. The second one will show you how to analyze these simulation results in different ways. Then the third one is on root hydraulics within the root. And the fourth one is including also the dynamic soil example. And so I hope that maybe you would be interested to try a couple of those. So to make it easy for everyone and to avoid different hardware compiler settings, I prepared it for Google Colab. So all you need is a browser and internet access. It helps if you have a Google account as well. And then you can go to this GitHub repository and start your own simulations. And with that I would like to finish, and I'm happy to take questions.
Okay, the questions are one more comment following up on Ruben's question and on the interactions between different groups. You can say that we will get more and more into understanding the single group, but I think we have some examples that sometimes the interactions is more important than understanding the single one, like in bubbles of gas in water or droplets in air, the disease spreading. We the interaction or the competition is very important. So kind of, in a way, it's good to invest in that, not just in the specific single route. It can overshadow
Well, you mean the interaction between different plants, right?
different plants.
Yeah,
yes.
because the roots are interacting. The individual roots here are interacting in this simulation that I showed you. So we can also do multi-plant simulations, right? But at the moment, the interaction is, as I said, indirectly. So all of the soil conditions that are changed by neighboring plants will affect the other plant. But yeah, so maybe you are aware of the exact mechanism, maybe some kind of signaling or so. What makes how does this interaction on the mechanistic basis actually occur? I don't know, because that for me would be the next step to look into this. I agree it's important. It depends also on your scientific question, whether you are in an agronomic experiment or if you are more interested in ecology with different communities, and there the interaction is of course more important.
And just a quick question, how fast is the genetics, kind of the last point that you showed, the genetic changes in
Yes.
relation to the condition?
How fast?
The time scales for these events.
Yeah, so well this was...
In one plant life?
No, so that was a work that looked at 8,000 years ago until today. So I can show you that, or I can let you have the paper if you want, because then we have these. So more than 3,000 cultivars were also genetically analyzed in this paper, and they came from different times. And so we have kind of a list of when did which variety appear. And so we didn't follow, so we followed it after. No? So we looked at it, so this line now appeared, how is its root architecture compared to the climate. So we wanted to make this relation between climate and root architecture. And the root architecture, as I said, it's not on purpose, never was bred for, right? So the first domestication from starting 8,000 years ago to now, the first time starting, say, 8,000 years ago, it traveled just, right? So it was not even active breeding, or probably not, I don't know what 8,000 years ago, if we can talk about active breeding then. But it was more like indirectly somehow that it adapted and a more success. So that's how I envision it at least. Later we have active breeding programs, right? Where plants were selected for, but they were selected for based on yield, and not based on our information of the root architecture, and so we were interested in this fact. But if you're happy to look at, I'm happy to show you these different timelines of these different cultivars.
Genetics like the equivalent of a very short time scale, small changes, not in the actual genetic information, but the manifestation of that.
Yeah, so well, I would have to look that up. So I didn't look at the time scale actually. So I just know that it continuously changed from 8,000 years ago to now. When did the changes actually happen? We can look it up together if you want. Yeah.
So
I don't know who is.
First of all, I would like to thank you for a fascinating presentation. I have many questions. I hope we have a chance to discuss. I will choose just one question. To what degree does the root architecture influence water movement during, say, infiltration events? So I think you didn't discuss much the infiltration events, how those are modeled.
Yes.
You mentioned Richard's equation, which
Right.
I have my own personal opinions about.
Yes, sure.
Besides
Me too.
the question here is more, does the root geometry and architecture influence that downward movement of water during filtration, in your experience?
Yeah, actually it does, yeah. And we also do at the moment some simulations for soybean and maize fields in the US Corn Belt. And there we simulate what is the difference if we have roots growing in this field or not. And and we look at the water movement based on including all the climatic data, also the infiltration coming from precipitation or irrigation, and also looking at the nitrate transport within the soil and how it and then we are also interested in placing the perfect timing of, for example, a fertilization event with regards to the climatic development, and we can simulate that in some cases the infiltration. So when, or an interesting question is, when does the infiltration go below the rooting zone and is lost, so to say, as from an agronomic perspective, maybe not from a groundwater recharge perspective, or the nitrate. And so that makes a difference. But now your question is whether the different root architecture makes a difference, right? And I would say yes, it does. So we have, for example, a big difference in deep rooting versus shallow rooting architecture. I also made the experience, though, that kind of getting the overall boundary conditions right is the first step and has much stronger influence. So the eco-hydrologic conditions of a specific site, for me, that is you have to get this correct first, and within that you can then have the differences that could be attributed to the root architecture differences. I think this is really interesting question, because now all the breeders come to us and they say, yeah, we want to breed, which root architecture should we breed for? And now that we are doing the simulation, we're looking exactly at this question. So how big is the effect versus all of the other effects we have on the hydrologic conditions of the overall site.
So in your model, mechanistically, do the roots act perhaps as preferential paths, or is there?
So you're talking macro pores and preferential flow, or
redistribution through the roots.
Through the roots.
Yeah. Well, that's what I tried to say. Yeah. Well, so first of all, yes, through the roots, yes. So there is redistribution through the roots. This comes naturally out of the formulation of our model.
Yes.
And that's what I try to show with this 100 or 10 years versus weeks. So that is part of the model. There, I think that plays a big role. But that's more once the soil has already quite dried out, well, then it's an important phenomenon. If it's anyway a moist setup and then you have additional irrigation, then redistribution is maybe less important. Now, if you want to talk about preferential flow within the soil, well, so our model is kind of a modular setup where we can connect to any kind of soil model. So I only talked about simple Richards equation simulations. Of course, we can also connect it to any other flow model that computes also preferential flow, for example. But there we didn't look at the effect of the, so what's more important? Preferential flow versus route as hydraulic highways. All right.
maybe you can discuss it.
Do your models account for the role of ectomycorrhizal fungi in bringing water to roots, so that they have like deeper access?
Yeah. So very good question. So I didn't talk about this topic at all, but actually I have had several years of research and model building in the topic of but arbuscular mycorrhizal fungi, actually. And so the short answer is these models where we do the root water uptake, there we don't explicitly account yet for any mycorrhizal impact, other than perhaps indirectly if they change the soil hydraulic properties. Because historically I've been focused in the topic of mycorrhiza more on phosphate uptake to plants and how the mycorrhiza helps the plant in phosphate nutrition. And there I have done several models where we simulate the root architecture development, the infection of the growing root architecture with the arbuscular mycorrhizal fungi by primary and secondary infection, then the growth of the external hyphae into the soil, and then I only computed the phosphate uptake by the hyphae and then by the plant. I'm currently part of a project that now looks at the effect of mycorrhiza on root water uptake. So, but there's no results out yet, so I'm sorry.
So the graph network approach for the root architecture is really cool. I think I missed it in your explanation. What were the mixing rules for when two edges intersect? Is the flow unidirectional to the base of the plant, or can it go into different roots? And what was the reason for how to choose this?
So the flow is only ever in according to the the xylem water potential. So that means the water potential gives you the way of the direction of the water flow. So it can potentially move in any direction. So because the flow is described so on a segment basis between two segments, between two nodes of the segment. And so there's no mixing as such, but it's just a simple flow equation according to the hydraulic gradient. Of course, then on the overall root architecture level, there we collect the flow in and out of each of the nodes by Kirchhoff's law.
And so can it go into like other roots, or does it?
Yeah, it could. Well, it could potentially, but most of the time it goes up, right? Because the transpiration pool will make it go up. But in theory, it can go in any direction.
Okay, so left side no question. Okay, go for it.
I wanted to ask whether you perform some kind of quantitative comparison of the simulated root architecture and the real roots, like, I don't know, measuring the angles of branching or distribution of diameters.
Oh, that's a very big topic. So how much time do I have? Okay, so there are currently, in our community, when we apply for a research project, there is almost always some way of root observations, some kind of root observation part of it. And we broadly distinguish between lab-based approaches and field-based approaches. The lab-based approaches, we have all kind of methods, like, for example, these, what I already explained, these rhizobox experiments. There we have in Jülich this plant phenotyping facility where really large amount of plants are automatically scanned daily and how the root architecture develops. From that you can extract all kind of information about the roots. We also have imaging of three-dimensional imaging of roots. That's then restricted to rather small plants, maybe up to 3 weeks old plants. We can put them in either an MRI device or a micro-CT imaging device. And there is also a lot of work going on in terms of image analysis, what's the recovery rate of the roots from such an image, because you lose the smallest roots, and how much is that, and so on. And also we have kind of VR goggles by which you can then virtually go in 3D, walk around your root and kind of make manual drawings and do all kinds of things you want. And also that with the goal to extract quantitative information of the elongation rate, of the branching angle, of the branching distance, and so on. So, but of course these are only young plants, and there's always this debate, how well can this be applied to a field setup. And so then we have also the field observations, and they now mostly do either coring, so you just hammer the soil cores in the soil, and then you have you divide the core into layers, wash the soil from the root, and scan the roots, and so you have root length density profiles or with depth. And then we have the rhizotube approach, which I showed you a little bit in this video. So we have really a field site where up to 120 centimeter depth, we have these long transparent tubes installed, and we can observe the root growth by cameras. we then get things like root arrival curves. So we know at which depth, when do the root arrives, and when do they reach their plateau there. People also do trenches. They really excavate, come with a big machine and excavate like the trench, and then count on a grid-based analysis how many roots per grid. And by this way you can also get a better picture of the horizontal lateral heterogeneity of the root distribution. And we try to use inverse parameter estimation algorithms to infer root architecture parameters from such data.
Andrea, that's a delightful talk about a very elegant model. I have a lot of questions like Ruben, but I'm going to ask just two. Number one, does the plant functional type figure into your model? Because that's the connection with the land surface community.
Yes.
They classify plants by functional type.
Right.
Herbaceous versus woody, deciduous versus evergreen, for example. Does that enter into the model?
Well, that is exactly the level of discussion we are trying to do at the moment, because because current plant functional types, as far as I'm aware, have a rather coarse way of describing the roots. And so we wouldn't enter the plant functional type in our model, but rather we would like to enhance the root description in the plant functional types in the land surface models based on our approach. And for example, what we could do is compute KRS and SUF. So these are really only two things, yeah? And we could compute them. That's actually an idea we are trying to follow up now for these different plant functional types. So we would have to find kind of representative plants for each plant functional types, and then we can compute KRS and SUF, and this then could be added to the land surface model. So that's the general idea.
You can, from below ground traits, hydraulic traits, you can infer, what is the above ground. Can be a different way of classifying plants based on root hydraulics. That's one question. Second question, do you have carbon limitations?
Also a very good question. Okay.
That's how you connect to the above ground, which is really beautiful idea there.
Yeah. So the first models, one of the ones I showed you that only simulate the root architecture, they only grow according to growth rules. And that's kind of the potential growth if there is no carbon limitation.
There's no carbon, water, nitrogen limitation.
So that was the starting point. So we have now developed in two directions regarding this. The first one is to couple our root architecture model to crop model, like Simplace. And there we receive the information of the available carbon in each time step from the crop model, and then we compute the potential growth based on our growth rules, and we compute based on some assumptions of the tissue density and the carbon for each tissue which is required. We compute the growth according to the carbon availability. And then another way we extended our model was to expand it into a full 3D plant architecture model. And so we simulate there also the photosynthesis, and we follow the carbon into all of the processes. So the carbon is produced, then it moves through the plant to the different organs, where the organs can use it for maintenance or for growth. And then, of course, it's carbon limited, because only the carbon that arrived at this specific organ can be used for growth. So that's a difference between the crop model and this approach, because the crop model has the overall availability of carbon in general and then distributes it according to some coefficients to above and below ground, which you have to prescribe as the user. While in this approach we don't need to prescribe it, because the transport, the photosynthesis itself and the transport through the plant will limit how much carbon is available in each position within the plant. And then if there is then still carbon left over, it can be released as rhizodeposits.
Makes sense. Thank you.
Okay, so any more questions? So if not, we thank speaker again.